we've got new ideas flowing in quickly. We're shrinking the time of development. We're not the only ones that have drugs for undrugged targets. There are other groups that are starting to fill that pipe. So I think the next decade is going to be very exciting for those who can make sense of the programmability of the cell to take all of these new tools in the toolbox and think about, okay, where can I apply these? How do I get precision medicine to say this patient population needs this molecule or this cocktail of molecules to restore its homeostasis, right? So if we can do that, I can see, everything from small molecule sets that can trigger Yamanaka factors for partial reprogramming, and some people are working on that. Maybe that's the answer to this right set of molecules can stop progression of Parkinson's or Alzheimer's, and like we've now eliminated a huge core unmet need for aging people.
Hi everyone, welcome to episode 18 of The Polling Principle. I'm here with Andy Lee of Vincere Bio. We're excited to have this conversation today. Thank you so much for joining me, Andy.
Yeah, thanks for having me. Looking forward to it.
Cool. So I think we met maybe a couple of years ago at ARTD or another one of these conferences, and this year's edition of ARTD just finished, right? As of the recording. Did you attend? How was it?
I unfortunately couldn't make it to this one. Had a few other commitments this week, but it looked like a great one. It just gets bigger and bigger every year.
Yeah, I think they're just going to run out of space on the University of Copenhagen main hall, right? That's kind of the limiting factor. Cool. Yeah, no, but I was tracking a little bit the news and it seemed very interesting. To begin with, I'd love to hear a little bit about yourself and especially your path into, longevity biotech. So, please.
My background originally is in software engineering. I started coding in elementary school way back in the early 80s, and then went through college for that and went into a couple of industries before finding biotech. But along the way, I had the fortune of marrying a brilliant neuroscientist. And so Spring was working on Parkinson's disease. And we started to realize she saw this human data that was sitting underutilized while I was seeing NVIDIA GPUs coming online to support a really massive compute. But the biologists didn't know how to query the data, and the computational people didn't really know what questions to ask. And so we were able to join forces and kind of bridge that to build some interesting simulated biology systems and systems biology to predict better drug targets. Also initially starting to try to find a cure for Parkinson's disease, but Parkinson's led us to mitochondria, which is of course a hallmark area of aging. And here we are now with molecules about to go into the clinic that can rejuvenate mitochondria.
It's very interesting, and hopefully more people coming from the engineering or from the software engineering world will start thinking a little bit more about longevity. I think for many years it's been so hard, right? Because the longevity field or the biology field in general is not super welcoming to engineers. There was this anecdote that I just tell, like you could take literature graduate and then put them on a 3-month boot camp to learn JavaScript, and then suddenly they are a front-end developer, right? The software engineering field was always very open to people coming from outside of the field. But imagine, taking a software engineer, putting them through a 3-month, reading biology of the cell, right? And then saying, "Oh, now I'm a biologist," right? Like that, it would not make any sense, right? And I wish it was a little bit easier for folks, coming from software to, do something interesting or meaningful in biology or in drug discovery.
Yeah, I think it might be getting more to that point. The last few years of generative AI have really changed the game. I think a lot of what used to be walled garden knowledge is at the fingertips of everybody. And so critical thinkers who have some background in how to problem solve can now find a problem they care about, like aging or disease, and like really rapidly iterate on getting questions answered and generating the next interesting questions. So I think we are in a place where software engineers or technical people might almost become product managers in the biology space. They're still not going to go put their hands on the bench or run cell studies or mouse studies or these other things, but they can really come up with the ideas of where the science should go, and then pair up with those technical folks who can go into execution mode.
That's interesting, and it just came to me as you were speaking. Maybe the limiting factor is not so much the technical knowledge, but the business model, right? Like in software, you can go and say, "Oh, I'm just going to build a tool to let people download a PDF file of their bank account, and then put a Stripe checkout page, and then charge them $10 a month to use it, right? And then you can start making money out of it." And in biology, there's no good way to do that, right? Like especially in drug discovery, you make an investment now, you get paid hopefully 15 years from now, right? It's really really hard to bootstrap something.
It is, yeah. I was actually just this morning reading a new article that Lada wrote, talking about why there's no $1 trillion biotech company, and the business model is a piece of it. It takes, everybody quotes $2 billion in 20 years to get a drug to market, and we've been talking about this a lot lately, that there are some rigid bottlenecks and some flexible bottlenecks in the process, and so we've been able to apply AI and efficient, workflow management to shrink some of the pieces, but then you still get into, even with robotics and AI, designing, synthesizing, enzymatically testing, running through the panel of ADME tests for every molecule in your pipeline, still takes weeks or months. And if you've got to iterate 1,000 times to get a dev candidate, that's just going to take years to get through. And when you get to human clinical trials, if you want to empirically show that you're slowing progression of a disease like Parkinson's, or even worse, slowing aging itself, you're going to have to track for 5, 10, 20 years before you can actually say, yes, my drug really worked. So, I think there might be some things emerging with better business model shifts. Like if the FDA gets out of trying to approve for efficacy and just approves for safety, and then the market can, judge on efficacy, that's something I've heard floated that could be interesting. That would allow you to go to market after phase 1. Maybe you start to make some revenue that works. The other path that is kind of more in the traditional realm is thinking of it as a relay race. So if you're doing early stage invention and optimization, you should probably plan on handing off the baton around phase 1 or IND, so you can, you'll cash out something, probably not, the tens of billions a year that a drug like this might yield, but you'll be able to totally de-risk your, smaller investment, smaller payoff, keep the potential for royalties on that might come down the road, but let a partner that is running at a different scale take that clinical stage work for the next 10 years. That keeps people a little bit more, cuts it in half at least, right? Where you can now iterate on 10-year projects instead of 20-year projects.
Yeah, I think that's very smart. And my recollection from conversations that I had a while back, kind of around the JP Morgan conference, was that maybe much of early stage research is going to be outsourced to Chinese CROs, right? A lot of the animal experiments, a lot of the chemistry would be outsourced. Maybe the innovation around the novel target, for example, would be done in the US, but then everything else would be done in China, and then somebody would take it to IND studies, and then once it's approved for IND, it's sold to a European pharma, right? It's going to be maybe a little bit more of a I don't know, an ecosystem, right? Like I guess in manufacturing, right? Manufacturing just go and, you come up with your, cat piece, and then, you outsource everything else, right? And then, you market your product, right? And I maybe, yeah, like the older way of, trying to take a drug to market, it's just going to slowly disappear.
Yeah, it might. I think that sort of global collaboration and efficient operation is almost table stakes at this stage. Almost all synthetic chemistry is happening in China or India at this point, and trying to rebuild that infrastructure in Europe or the U.S. Would be very expensive and difficult because the skill sets are just not here. Like we don't have the people to do that sort of thing, and robotics are not quite there to do it at scale. And then the other part that I think is interesting is does that carry through to the clinical trial approval and the business model stage? Can you also do similar, things where as soon as you've got some reasonable demonstration of preclinical safety, can you take it to market through plates like Prospera, and put things on the market before they've gone through any FDA type trials? Do we still have a responsibility to do some of the same GLP safety studies, GLP manufacturing before we enter that sort of thing? I think probably, like as a manufacturer, I wouldn't be comfortable taking and on. There's a level of evidence that I would need before I could sell this to humans, but that level of evidence is somewhere short of 10 years of gated, placebo-controlled trials.
Yeah, I mean, I think that, maybe that's one way to get a drug approved and get some kind of staged revenue before you can get it approved in the US, right? Like if you could get your drug approved in, I honestly, I don't know, like what would be a country with maybe easier approval process and high population, like Indonesia or Brazil or whatever, but like, if you could get it approved that in 3 years, get some revenue, and then eventually work towards a large phase 3 and approval in the US, for example. I don't know, maybe that would be one option, right?
Yeah. Yeah, I think that could work. The other interesting wrinkle that's different in drug development as opposed to like software is the how easy it is to copy most of these things. Like once you have published patents or molecules that are proven, I think the IP protections really are what give people the defensive position to capture higher than generic prices for at least a decade or two, as opposed to the software world where you, you have a lot more trade secret, you have a lot more brand driving the value, things like that. Or maybe the drug world just also needs to get in this mindset that you're going to have to replace your drugs every couple of years. If we get faster on innovation, maybe that would work out.
Yeah. Yeah, in some ways, it's a little bit crazy, right? Like the drug world is open sourced by legal requirement, right?
Yeah, yeah.
Cool. Okay, now let me switch a little bit gears a little bit, so we don't get lost, so we don't lose the biology. So, why mitochondria? I guess a lot of people would know like the energy from the cell comes from the mitochondria or whatever, but mitochondria do a lot of other things. Well, like why were mitochondria interesting to you or Spring, and like how did you get into it?
Yeah, it's actually a really interesting story. We started by stepping back with an unbiased approach for Parkinson's, and instead, Parkinson's disease is this interesting space where there are about a dozen genes that have Mendelian inheritance for the disease. If you have, one or two copies of one of these mutations, you have very high penetrance to get the disease. But they're in different pathways. So a couple of them, PINK1 and Parkin, are directly involved in labeling damaged mitochondria for mitophagy. So that's been fairly clear, but those patients get really early onset Parkinson's and often don't even have alpha-synuclein protein aggregates. So the field for a long time said those are just not even really normal Parkinson's. They're something completely different. And then you've got things that are involved in lysosomal function or trafficking, other things around the cell. So they seem somewhat disconnected. But we built a protein interaction graph that allowed us to kind of look at the overlap of those genes. If you think about the network of interactors around each gene that can cause the disease, and then you look at where those networks overlap, you can find nodes that have the highest centrality and kind of rank order by that to figure out what might be common drivers of the disease as well. So when we did all of that and then ran through some ontology analysis, mitochondrial quality control was consistently the highest overrepresented point of convergence. It was the pathway of highest impact in that gene network. And that even held up when you started, if you leave out genes. So we did a leave one out iteration on that, where if you take out Parkin, still mitophagy is the highest ranked area. So that's what really gave us conviction that this is probably not just a pink Parkin problem, it's an all of Parkin problem. We then used our virtual cell simulations to take omics from sporadic Parkinson's patients and created simulated dopaminergic neurons using human data. And we found that those virtual human neurons were deficient in mitophagy. So now we had evidence that even the sporadic non-genetic forms of Parkinson's were deficient in mitophagy. And that's when we said, okay, this is going to be a big enough potential, it could be all of Parkinson's disease. Let's see if we can find a molecule that could do that. And then, of course, as we got into that, we started to hear about longevity a little bit more, connected up with Aubrey. And, he's, of course, very big on the idea that dysfunctional mitochondria are kind of a driver of the loss of ability to repair the damage up to mitochondria, particularly being a problem. And of course, now we've done collaborations with some other groups, and we've shown our mitophagy enhancers, they can accelerate recovery of acute kidney injury, which is something that also fails with age. They reduce occurrence and severity of abdominal aortic aneurysm, which is a completely different system, but where you've got age-related and metabolic drivers. We reduce senescence in cardiomyocytes. So as you would expect, this shared mechanism of aging does have benefit beyond just Parkinson's.
If I understand correctly, mitochondria have a life cycle, right? They when a cell, splits, it comes with some mitochondria, and then more mitochondria come up, right? Because the current mitochondria that are in that cell, they split, and sometimes they fuse again, sometimes they get recycled, right? And do we understand how that cycle works? Is it based on how many there are, or the type of cell, or what triggers those mitochondria from what triggers them to spawn or to be recycled? Is that already understood?
Yeah, it's really fascinating and a lot more dynamic than most people realize. The mitochondria do constantly fission and fuse into this big network. There is biogenesis of new mitochondria along the way. And when mitochondria become damaged, which happens normally over time or through events, there's a process called mitophagy that labels the damaged ones for clearance. This process is really well understood now. In about 2010, Richard Ewell from NIH published very detailed mechanism of how this happens, and it's coordination initially of accumulation of a kinase PINK1 on the outer mitochondrial surface that phosphorylates ubiquitin, which recruits Parkin, which is an E3 ligase that again gets phosphorylated by PINK1. That really rapidly adds chains of ubiquitin to the outside of the mitochondria, which recruits the autophagosome machinery to engulf the damaged mitochondria, take it to the lysosome, where it gets broken down into parts that can be used for building new ones. That also triggers an upregulation of PGC1 alpha so that you get new biogenesis.
So pretty. It's said mitochondria is big, right? It's not like a tiny little thing that you can, I guess, put inside of a lysosome, right? Like how does it get engulfed by the lysosome?
Yeah, one of the first things that happens is the damaged portion of the mitochondria gets fissioned off from the network. So it has to become like a smaller chunk that's free floating from the main bit, and it gets untethered from the cytoskeleton, so the trafficking of it stops. So these are usually like running around on the cytoskeleton, and so you've got this multi-step process of break it off from the network, stop it from moving around, label it with ubiquitin chains. Then that's a small enough chunk that can get wrapped up in an autophagosome, and then that is structured in a way that autophagosome gets trafficked back to the lysosome, where it gets fuses in and at that point.
Cool. And then, so it's the assumption that the more of this process that you have, the better, or is it like too much mitophagy? I guess like if your cell is constantly recycling mitochondria, that's also maybe a necessary energy usage, but it's not necessarily a bad thing.
Yeah, it could be. So, a lot of mitochondrial toxins depolarize mitochondria, and that causes an increase of mitophagy, and that's no good. And there are some mechanisms of increasing mitophagy that might just clear all of the mitochondria, which of course be cytotoxic and would be no good. But USP30, the specific enzyme that we're inhibiting, is kind of the brake to the process. So by releasing the brakes, you can accelerate it, but you can't actually over accelerate it because you're not stepping on the accelerator of the process. So what we find is that if you give our molecules to healthy cells, they don't do anything. If you look at enzymatic activity, they don't increase mitophagy to a healthy cell. But if there's mitochondrial damage where the cell's already inducing some mitophagy, then our molecules increase the rate of mitophagy above that. So I think that's a really important safety feature that, we're not going to wipe out the healthy ones. Because of course it's a balance, right? You want as many good healthy mitochondria as you can without, while getting rid of the ones that are spewing reactive oxygen species and other problems.
Yeah, that's very interesting. When I was when I started to learn a little bit about biology, this was one of the things that I found very fascinating, right? Like when you give some small molecule to a cell, you're basically kind of breaking things, right? Like you're throwing a wrench inside of something, right? Like, a small molecule that binds inside of, kinase, you just prevent that kinase from working. That's it. And most drugs are like that. They're just basically, throwing little things that fit inside of pockets and then prevent it from working. And then you think, well, then you could use that to prevent the cell from doing things that it's not supposed to do. But how do you get it to do the things that it's supposed to do, right? Like kind of, promoting things, right? And then I learned, well, you know what? As it happens, I guess we're lucky in biology, there's all sorts of like hermetic kind of cycles, right? Where, the cell will do something and then it will do the exact opposite, right? And then those two things happen in parallel, that sometimes one of them takes over, sometimes the other. And then if you figure out, which one is the opposite of, what you want to enhance, right? Like in this case, just, push on the brakes a little bit more, right? And that has the effect of what you have, right? And I guess we're lucky, right? Because I think it's 70-80% of approved drugs that are small molecules that basically just bind into things and then prevent them from working.
Yeah, that's a really insightful point that I don't think a lot of people think about. And it really came clear to us when we were building our virtual cell models that there's a yin and yang to every process. If you've got a kinase, there's a phosphatase that removes that phosphate that it's adding. If you've got GTPases or ubiquitinases or ligases have deubiquitinases, and So all of those. So often, one of the other programs we started actually before USB 30, we were trying to get activators of Parkin, which is the E3 ligase that adds ubiquitin. And of course, activating enzymes is difficult. And then also, even if you do successfully get a molecule that can increase activity, there you might have risk of overactivating that, because now you're stepping on the gas. How does the Parkin that's now constitutively active know to leave the healthy mitochondria alone? It really doesn't. It's just now going to do its thing. So I think that's where kind of stepping back and taking a more rational systems biology view at, okay, what's the end goal biology I'm trying to accomplish? What are all of the players that are around that biology? And then what are the most druggable nodes that can cause that outcome that I'm ultimately looking for? That's kind of the approach that we take to find targets. And, when you do so, you're most of the time not going to hit the popular genetically validated target that everybody knows about, because most mutations that cause disease are loss of function mutations. So you would want to be activating to overcome that. But it is exactly that, where if you've got loss of function causes something, maybe you can inhibit something else to restore the homeostatic balance between those two players, and get the cell back to a normal function.
Yep. Maybe veering a little bit of, let me ask you something a little bit more controversial. So within longevity itself, right? Sometimes I think people don't like small molecules because they think, well, with a small molecule, you're basically, inhibiting one of those things, and you're just, in an ideal case, you are restoring that homeostatic balance to the cell. But that homeostatic balance, is going to make the cell, die within 80 years, right? That's where we are, right? So if you want to drastically extend the life of the cell, there's no way around new genes, new quality control mechanisms, new functionality that you basically have to, write the code for in that cell, inject it in that cell somehow. I'm not sure I buy that argument, but I see how it would make sense, right? Where would you stand on that sense? Can we meaningfully not just cure diseases of aging, but also cure some diseases of aging, but also meaningfully extend healthy lifespan through small molecules, or going to need, genetic engineering?
Yeah, biology is inherently reprogrammable, right? And we see this in different cell types that have the same DNA but have different expression levels that drive totally behavior within the tissue levels. And you see it in natural reproduction, where you can take an old egg and reprogram it back to a bundle of stem cells that are epigenetically reset, so that old, mother cell has, all of these accumulated DNA damage and short telomeres and dysfunctional mitochondria and deficient mitophagy, and all of a sudden has a youthful state that then for the next 40 years can get progressively stay healthy and homeostatic. So that tells me that there is an ability to dynamically change the behavior of the system. We know how to do that, of course, now with transcription factors and Yamanaka factors to do that at that scale. But small molecules are an effective way to interact with the system. We know that we can engineer molecules or peptides or whatever that will bind to some receptor or to a specific enzyme and either activate it, inhibit it, or glue it to a complex, or all of these things that. So the limit now is to understand what is the interaction of all 25,000 genetically encoded proteins and all of the other lipids and metabolites in the system. It's big, but it's finite. And then within that, build more molecules that can act on that system. Right now, the approved drugs only work on, what is it, about 600 different distinct targets that are actually drugged by all of the known drugs that are approved. So there's a huge scope of undrugged but druggable targets. And so that's a
Quoted out of like 20 to 30,000. Yeah, sorry for like people that don't know that. Yes, please.
Yeah. There's about 25,000 genes that are or proteins that are encoded by genes, and we've only figured out in all of human history how to drug 600 of them. I think that's an incredible opportunity, particularly in the age of AI, where we can now do really understand some of these larger data sets and put together pieces into interesting graph networks. There are ways that we can tease those out. It also brings along challenges. Like, I can tell you from experience now, when you try to make drugs for an undrugged target, the pharma and regulatory worlds, probably rightfully so, are very cautious to see, okay, show me evidence that it is actually safe to inhibit this thing. Show me multiple species, check all of the tissues, show me for a very long time, right? So I think there does have to be some caution as we add these things in. But I think the world is starting to prime that pump. Like, we've got new ideas flowing in quickly. We're shrinking the time of development. We're not the only ones that have drugs for undrugged targets. There are other groups that are starting to fill that pipe. So I think the next decade is going to be very exciting for those who can make sense of the programmability of the cell to take all of these new tools in the toolbox and think about, okay, where can I apply these? How do I get precision medicine to say, this patient population needs this molecule or this cocktail of molecules to restore its homeostasis, right? So if we can do that, I can see, everything from small molecule sets that can trigger Yamanaka factors for partial reprogramming, and some people are working on that. Maybe that's the answer, to this right set of molecules can stop progression of Parkinson's or Alzheimer's, and like, we've now eliminated a huge core unmet need for aging people.
That proteins, sometimes people that are into biology, right, like, think of them sometimes in terms of like, they perform a single function, right? As in, in the software engineering world, when you have a little routine or a little program, and the program performs a function, and you have an API for it, and that program does not interact with other things, right? If you have a Linux program that takes a piece of text and removes a character out of that text and outputs that text, that program, you can put it in a directory with other programs, but it's not going to do anything, right? The only thing it does is what it's supposed to do. Whereas proteins many times are just, hanging out in the cell, or they're present in form of complexes, which are, aggregations of proteins, bound in different ways, and they have different functions, right? Like, they may have one function on one side, another function on another side, depending on the region of the cell, depending on what other proteins they're associated with, depending on what else is around them. They may have different functions, and nobody has gone and systematically said, well, let me look at all the, protein interactions and then figure out, what these proteins may do, right? But like you said, it's finite, right? Like, if you have 25,000, you could just run the, 25,000 times 25,000 matrix and figure out, is there something there, right? It seems like, somebody, with a few $1 billion worth of compute capacity should just, go ahead and start running something like that, right? And there would be a gold mine of, biological, insight to discover.
Yeah, it's interesting that that's actually precisely what we did 10 years ago to try to build, in order to do cellular simulations. There were two pieces of it. There's the spatial temporal physics of how do proteins move about a cellular environment, but then more importantly, what happens when two proteins bump into each other? And while there are libraries like BioGrid and Intact and Reactome that have libraries of these two things do like co-IP or bind to each other, there wasn't a good data set of deterministically what happens on the other side of that equation, right? And so that was one of the things that we had to curate through a series of data mining and manual efforts, but building that library of if these two things bind, they, hit, they're going to bind. If anything else hits, they're just going to bounce off and nothing happens, right? If in order for a kinase to phosphorylate a substrate, it has to first be preloaded with an ATP molecule so that it has a phosphate to transfer, and then that'll let off an ADP, right? So you can break down cellular biology to that really granular deterministic set of rules, but you have to also keep in mind that some of these proteins are kind of like little Swiss Army knives. They have multiple functions depending on, they'll do one thing with, LRRK2, which is a gene where mutations cause Parkinson's, is a great example. It has kinase activity for some substrates, but it also has GTPase activity for other substrates. And when you have LRRK2 G2019S mutations, it increases in affinity to kinase phosphorylate some things, but decreases affinity to phosphorylate other things. So it's not nearly as simple as this thing causes the disease, let's just inhibit it, right? You really have to think about which thing. And a lot of these proteins, the state, the functions they perform is driven by a state that is also knowable by phosphorylation on different sites of the protein, which, molecular biology wise changes its shape, which then opens up pockets so it can do other things. But even if you just think of it as a simple state machine, that like if each biochemical entity has some set of function spots on it that can be called, that would then set a state trigger that would then say, now I am primed to do these three activities, and then I get these phosphorylated. Now I can only do one of those activities. Right? So it's complex and it's highly dynamic. But at the end of the day, you've got 25,000 proteins that each have some, 1 or 3 or 10 states that they have that will perform set of actions. Those actions might be limited to, I do these things inside the lysosome because there are pH sensitive changes to the shape. I do these different things outside of the lysosome where there are, a different pH level, right? So you've got to think about compartments. You've got to think about proximity to other interactors. You've got to think about state of each of the interactors. It's a very complex problem, one that was completely untractable before NVIDIA GPUs allowed us to do highly parallel computations, but that is becoming more tracked. The scale at which you can look at this problem is getting bigger every day.
Let me go run through another question that came up as you were speaking before we forget about it. So we're talking about like tectonic factors and so on, and as a way to maybe regenerate, cells or tissues. Some organisms do have the capacity of regenerate tissues, right? I think salamanders are maybe like one of the most famous ones. Right? You can take a salamander, cut the tail or cut a leg, right? And then it would regenerate the entire thing. It would be great if, humans could do that, right? Like, use an arm in an accident and just regenerate the whole arm. And I think, and this is actually very funny. I did not believe that it was true, but I looked up and apparently it's confirmed. Like human children under 5 years of age can sometimes regenerate entire phalanges of their fingers. So that is funny. Like, you would imagine, it's like, once the bone is done, right? It's done, but now there are multiple confirmed medical reports of children actually regenerating entire phalanges. But I think what is not clear, right, is whether the biology that enables these animals to regenerate part of the body is somehow conserved in humans, right? Like we have no idea to know, right? So, I wonder if that may be like people could be looking for that for decades, right? And eventually not find any kind of combination of, reprogramming factors that somehow triggers that regeneration. Like we just don't know, right? Like some animals seem to have it, some animals don't, right? And I don't think there's the knowledge of, when or how it's triggered is complete at all.
Yeah, that's super interesting. I hadn't heard that about children regrowing phalanges, but it makes sense, right? So some animals can do this. So if that's the case, we all share that same ancestry of DNA, multicellular animal function, right? So that should be possible. And then again, even within humans, going back to natural reproduction is a great example that, like, okay, inside human biology, there is some knowledge of how to regrow the whole template of all of the tissues and everything that you need. Uh, so I don't have any clue how you would do it, but it seems technically feasible that if you take partial reprogramming and you can get to stem cells, which are clearly a part of it. You're going to need to get a lot of stem cells into the place that can then redifferentiate into the parts they need. How do you steer that? How do you get it into the right place and not the wrong places? How do you tell the whole system that this is the part that's missing and needs to get rebuilt? That I don't know. And even like salamanders that can regenerate their tail. I mean, can they do all other pieces? Like how, if you chop off a leg, can they regrow a leg? So it may. It's one of the. I think back to the programmability of biology. At the end of the day, it's all just different combinations of proteins and factors that are behaving in some way to perform some function. So if you can induce the environment that tells that cell to behave in a different way, that whole population should start to proliferate into a directed manner. We just don't know what those factors are. Maybe they're bioelectrics, maybe they're chemical. A lot of possibilities there, but it seems finite and solvable.
Yeah, I mean, I sometimes think about this from the perspective of, so I believe somehow tackling aging is a multifactorial thing. Like there's not going to be a silver bullet, right? And then sometimes people say, well, yeah, cellular reprogramming would do it. And I was like, no, it won't, right? Like, let me tell you about, problems A, B, and C that is not fixing. And I was discussing this with, I think it was Claude or ChatGPT the other day, right? Like if you had, for example, a nerve that is lost, through whatever reason, right? Like, I think it was looking at, neurodegenerative diseases, but you can imagine, in trauma, for example, you go, you cut that nerve may be a meter-long cell, right? Like a single cell, it's a meter long, and then it's physically cut, right? Like, how would you even regenerate that, right? Like, so I was going through all the different problems. Look, one of them would be, well, there's a large energy usage, right? Regenerating a lot of that cell, would be very expensive energetically. You would need to figure out how to get that cell to actually regenerate, right? Like, that kind of reprogramming part of it. And you don't have, that cell grew in the context of an evolving organism, right? So you know how to reset from scratch, right? Like that's what, having offspring means, right? You reset from scratch and then the body knows how to do that. But without resetting from scratch, how would you actually do it? And then the really hard part is, well, how do you actually get that targeting to work, right? Like you want that nerve to, start, somewhere in the, spinal column and then, slowly find its way in between muscles and blood vessels and go through all the stuff and then, find its way, a meter long, from there and then bind to some muscle, right? Like that would be equivalent to like, well, you threw a rocket and then you land on, like a, 2x2 meter, squared on Mars, right? And like you cannot adjust along the way, right? Or like you somehow need to figure out how to adjust along the way. Like, there's no realistic way to solve that problem, today. Like we just wouldn't even know how we would start tackling that, right? So yeah, I think like reprogramming factors, even if like they will definitely solve probably a lot of things, right? It's just there's no way that they can be a silver bullet, right, for longevity.
Yeah, I have high hopes for reprogramming in some spaces, and I think being able to produce stem cells is a likely part of the problem. But if you have underlying deficiencies, like your mitochondrial network is out of whack, or you've got DNA damage in your cells, if you put stem cells in, or you've got protein aggregates, all of these problems are likely to propagate into your new stem cells, and over time they'll just become sick like the rest of them. The directing of axonal growth is another like challenge, and if you're going to put stem cells, where do you put them, and how do you get them to then reach out where they're supposed to? So I think there are, the cells naturally have some really exciting repair mechanisms that are there. Like, if you break an arm, it regrows back together. If you cut tendons, they'll like regrow. Even like some of these broken cells have ways to like fuse back together. And so I think if we can figure out like what are the factors to optimize that sort of recovery and repair, that would be helpful. I think there's probably some layer where we're going to have to get into tissue replacements. Like, I'm pretty excited about the idea of vat-grown hearts and kidneys and livers, so you can have your own there. Starting to pool your own young cells for later use might be a smart thing to do. So I think there's going to be some blend of organ replacements and cellular reprogramming, and partial reprogramming, and molecules that can adjust things back. That will be necessary to get us all there. But yeah, I don't see a silver bullet coming for like rolling back like stage.
And then maybe a combination of those things, right, will get us to like 200, 300 years. Who knows, right? And then at some point you'll be like, well, yeah, now we do really need to like write our own genes, right? Because the current machinery is will reach the limits of it. Who knows, right?
Yeah. I mean, at some point, like, it's one of the, this will be controversial for you. The death is bad, like longevity is the goal for everything, I think is somewhat problematic, because everything dies. Everything has, like, the universe hasn't always been here. Like, all things, the nature of any being is that it is continually like built up, broken down, recycled, brought in. We're constantly dying and remaking and preserving cells in our body, and then our entities within the larger ecosystem. So I think it's great to try to extend it as long as possible, but at some point, everything needs to be recycled. Maybe we can move our consciousness from one entity to another. That might be really cool. Like, if we think about what really matters in like longevity is, I'd like to be able to keep doing cool stuff forever. Whether that has to happen in this body, not sure I really care. I'm certainly open to replacing parts as much as I have to. And at what point have I replaced enough parts that this is no longer my body anyway? But as long as it's made.
Good enough. Yeah. I mean, 100%. And I think, truly a lot of people, like the longevity of which substrate, our software is running on, it doesn't even matter that much, right? Like, the only thing that people really care about is this continuity of consciousness, right? So, even if you were replacing neurons, right? Like, the brain is very plastic, right? So your personality may slightly change, but your personality changes anyway, right? Like, we all, age and, hopefully become wiser, right? Or at least, learn more things. So, at the end of the day, I think the only thing that most people would care about is like, do I feel like myself, even if my, my brain, my personality is slowly changing. I just don't think it's realistic to consider kind of the whole, transhumanist argument for the next, few hundred years, right? Like, it's just technology is not really going to get us that way. So unfortunately, we're stuck, trying to extend, the longevity of our biological substrate for the next century or two. And I mean, it would be great if we can do it, right? Other animals do it, right? I mean, that's why I tell people, turtles would live sometimes 150 years, whales, right? Like, some types of whales will live to 100 years, right? Great sharks will live to 150 years, right? So why not
Yep. Yeah, and one of the first books I read that brought me into the longevity world was Kurzweil's, How to Live Long Enough to Live Forever, and I think that sort of stepping stone approach makes a lot of sense. But I have a question for you. Thinking about like the substrate of moving consciousness to, now that we're starting to see digital twins and AIs that can read all of a person's writings and do a pretty good job at replicating their mannerisms and their thought processes and the way that they talk. At what point do you think that becomes a reliable copy of your consciousness, at least that up to that snapshot in time? And maybe we're already there.
Yeah, I mean, I think, in my mind, trying to duplicate how a person would behave is a much simpler problem than trying to replicate their consciousness. And I haven't seen any indication that it's possible to do that without having into like a minute level of detail, right? So like, even like when people would talk about, artificial neural neurons as like real neural neurons, but they're not, right? Like they are much, much, much simpler, right? Going from, a few weights to like the entire biochemistry of a neuron would be very hard. So my intuition is that it will not be possible to basically shift a human brain to run in silico until we can simulate the entire complexity of that human brain, possibly at the atomic level. So I if there's a way to do that without such compute heavy resources, I don't think anybody actually knows how to do that. So so if you think, well, what would it take, right, to simulate it at the atomic level? So you can say, yeah, we're running a simulation of an entire, all the atoms of the brain of this person. Like, we're not going to be there for many, many, decades, right? Even at the exponential increase in compute capacity that we see, right? So I think it's just a harder problem than tweaking the biology a little bit, right, to get an extra, decade here or there.
Yeah, yeah, definitely. Be interesting to see where all these things converge.
Yeah, but I mean, the really exciting thing, of course, I'm biased, right? Like the really exciting thing is like now that we have this technology, that is turning out to be useful, right? Like it can do a lot of things on its own, right? Like it can, hopefully start helping us, find new targets, right? And finding inhibitors for those targets or whatnot, all sorts of things within drug discovery. Now it's getting to a point where it's like, you have a human supervising it, and then, it performs tasks that are useful. And then maybe instead of the human spending, a week, the human can do it in 2 days, right? With the help of AI. But of course, once you get into any kind of exponential trend, like that, hopefully is uncapped, right? And you get to a point where like, well, maybe it's better than the human, and at some point it's 10 times better than the human, and at some point it's 100 times better than the human, and then what things could it do on its own, right? Like that's the really exciting stuff. Like if you go and throw, a disease at an AI and tell them, how do I solve this disease, right? Like, well, we get to a point that it will come and say, you know what, easiest way to do it is with like, this, genetic engineering that changes some, base pair somewhere and you have no idea what it's doing, but it would result in some cascade of effects somewhere else that would fix the problem. We're like, okay, well, no human would have got there, right? But the AI solved the problem for us, right? I think that's, that would be super exciting.
Yeah, the challenge is that, even if that AI system came up with the right hypothesis, that would still just be the first tip of the iceberg for this long process where, okay, now you're going to have to come up with a way to actually make that change in the biological system, in the right tissue of interest. You're going to have to show that it can be done safely. You can't trust that the AI didn't think about all of the other downstream effects of that. You're going to have to engineer a way to do that that doesn't have off-target effects. I think there's, which is still, in the big scheme of things, a 10 or 20 year development cycle to actually get something that works. We're talking about the rest of humanity that will benefit from this. So maybe it's okay as long as we can, start increasing our win rate and things that come out the other end are out.
Yes, we'll be there, right? I mean, like hopefully the jobs will be there as well.
right? If you start that's been one of the most interesting things of getting into longevity is being able to start thinking on entirely different time scales. Like I'm not going to retire in 10 or 15 years. I'm going to think about like, how can I keep compounding value for the next 100 or 200 years? And like, if something happens along the way that short circuits that, so be it, whatever. But I think that's a really exciting mind shift that you can start to do some foundational things and just have the patience to grind at it and work at it and say, okay, I see all of these new tools coming online. I see all of these new drugs that are going to become available. I see this reprogramming. I see these replacements. And like you can see that like the pieces are coming together in a time that will matter. And that starts to just shift the way you approach everything.
Yeah. Yeah. I mean, it is really exciting. Sometimes, people outside of technology, they just do not understand how fast things can go, right? Like,
Right.
what was it? Like the example that we put all the time, right? It was in the 1970s, right? Like, 55 years ago, the computer that put the man on the moon, right? Like, you compare it with like any, modern phone. Any modern phone is like, a billion times more powerful than that stuff, right? I mean, like, 40 years ago, right? Like we didn't have, internet and phones and this and that. And now we live in a world where like all the world's information is accessible, at our fingertips, right? So, I mean, if you go and say, I think the part that was always missing, right? And the reason, like, I personally got excited about working on tools for, for drug discovery, right? The part that I think was missing is that within tech, people understand the value of tools and building tools, right? Like you build your, assembly language, and then you build your compilers, and then you build your, your linkers and your libraries and so on and so forth. And that's what creates the exponential productivity curve, right? Whereas in biology, there's a little bit of that, right? Like, of course, people that worked on, CRISPR, right? And tools to understand biology itself, right? Like, the cryo-electron microscope and so on and so forth, right? But there has been a little bit less of an emphasis in, building the tooling that then, helps you, become a million times more productive, which I mean, hopefully will change over the next few years.
That's a very good point that the biology world, for being built on an academic premise of publishing your results and sharing your methods, really has been inaccessible and hard to use and hard to replicate. I think as this technology comes in, it will be important to make sure that the tools are accessible. That was one of our visions for our original computational biology, approaches, was to make these things in a way that biologists could do complex computational things without needing to be handheld by a computational biologist or software engineer. And it's a really difficult problem to like make these things where, somebody isn't just doing, really mundane, long configurations or having to have some understanding of like running a Python script. It's like not something that a biologist should have to figure out how to do.
Mm-hmm.
And so, what I'm kind of getting excited about now in the tooling space is being able to kind of wrap things behind these GPT interfaces that now everybody's getting comfortable with. And can we use that as how somebody describes what they want? And then the GPT can produce plans that an executor program can then like go off and like do things and start to, GPTs are great at generating code. Right now, they want a human to take that code and put it somewhere and run it. But like we can start to vet those in approved ways. Like we're going to get to a point where the AIs can just do a bunch of stuff and then say, oh, hey, by the way, human, I need you to go run this experiment that I can't quite do myself, or I need you to vet this plan. And I think that workflow is going to shift more and more automation, and a broader set of people will be able to just get involved, because that's ultimately what it takes. And I think we touched on this at the beginning, like how do we get more new people into biology? we have to make it as easy as talking to ChatGPT about what's the weather going to be like tomorrow. And you can start to explore instead, like, how can I, get better sleep? And like, that actually starts to go into workflow that might generate a new, like, hey, you need to take this new drug that's just, coming down the pipeline, and it's going to fix this problem because you described this other thing. I don't know. It's we can think about all the different ways that workflow could tie up, but I think there's a path forward there.
Yeah. Maybe like having an MCP server, right? When you call it and say, this is my molecule and this is my kind of experiment condition, run it for me and then get back to me with the results. It's kind of part of the reason why I really like what the Aura biomedical guys have been doing with the worm board, for example, right? Because like you can run one of those experiments and it's so cheap and so quick, right? I mean, like, I don't know, 30 days or whatever, right? Like, this is my molecule, go ahead, run it for me. And it may be like, I don't know, like $100, less than $100, right? Which I think, would, hopefully bring more people, into the space.
Yeah, that sort of hooking up, real world services, whether they be automated labs or humans that are now offering a service to do something. I think that's exactly right, Nate. Whether the AIs aren't going to just do everything, but MCP is probably the right interconnect or is the emerging interconnect. And you start thinking about how that can wire together. It's a very exciting time for software engineering. I know a lot of people are worried about software developers losing their jobs, but like if you step back into like, what can we do with this new world? It's a very exciting time.
Yeah, no, definitely. And I think we should probably leave it on that high note. We've already been speaking for an hour. I mean, this went quite quick.
Yeah, fantastic.
And I really do it like this because otherwise, like people don't finish the episodes, right? And I think this was a fantastic conversation. I enjoyed it very much myself, and I hope everybody else that might be listening to do well as well. But yeah, we need to leave it at about the hour mark, before people start dropping out.
Sounds good. Thank you, Javier. I really appreciate it.
It was such a pleasure. Thank you so much, Andy.